So good morning to all. Welcome to the Department of Microbiology and Biochemistry. I'm very happy to tell you that we have an eminent scientist from USA to give away the talk. And before going to that, I invite our head of the department, Gigi Jacob, to share a few words.
A very good morning to all of you. The Department of Microbiology and Biochemistry is very excited to welcome the participants from all over the world to this webinar on topic From Bench to Bedside, the story of how medicines are made by Dr. Robin Matthew. Dr. Robin Matthew is an internationally renowned scientist with several seminal discoveries on cellular metabolism and cell signaling to his credit. He has a master's degree in nuclear chemistry from Andhra University and a PhD in biochemistry from Baba Atomic Research Center. He is the winner of M.N. Shastri Gold Medal for the first ranker in nuclear chemistry. He awarded a scholarship from United States and continued his postdoctoral research in cancer metabolism at Princeton University. Dr. Matthew has over 40 peer-reviewed high-impact publication to his credit. He is the recipient of many prestigious awards. Currently, Dr. Matthew is serving as a global clinical strategy lead for Johnson & Johnson, where he has been the study director of several clinical trials, providing strategic leadership to the clinical development of new investigational medical products. I am very proud to have such an eminent personality for our today's webinar. Sir, I welcome you on behalf of Department of Microbiology and Biochemistry, St. Berchmans College. I'm very happy to say that we have participants from all over the world, including scientists, researchers, faculty members, students, and other professionals. Once again, I welcome you all to this webinar. Thank you.
Now over to Dr. Robin Matthew.
Thank you. Thank you for those kind words and the wonderful introduction. It's really such a great pleasure to be with you all today on the Saturday morning. I know it is early morning. It's a precious Saturday for you. It is Friday night for me. But regardless, please, can someone let me know if you can see my screen? Do you see it?
Yes. We can see.
Oh, okay. Yeah, thank you. So thank you for those wonderful introduction. It's my pleasure to be with you this morning, and I thank the SB College department, the organizers of this webinar, for organizing this. First of all, what I would really like to do in the next few minutes is to take you around the world on an interesting journey seeing places and telling stories about some interesting things. And then through that, we'll somehow connect to the topic of my presentation today, which is Bench to Bedside, the inside story of how medicines are made. So here we begin our journey. You are looking at a list of the world's best 50 restaurants. And it's not important for you to read the names of these restaurants. This is every year, there is a list that selects the best of the best restaurants around the world. And here is the first 50. Now we are looking at the top 24. And you can see some of the very famous ones over here. This is number 2, called Noma, that is in Copenhagen. Let's go to the top 12. Again, you see here Noma, and you can see some of the dishes a little more focused here. I want to take you to the top 3. And you see Noma here. And so here is a picture of the menu of Noma. The #1 restaurant in the world. I had the good privilege to visit Noma a few years ago. That was a wonderful experience. In the next one, this is another restaurant that is one of the best, called Martin Bichat. This is in Scotland. And you can see some of the dishes that is on the menu of Martin Bichat. I don't know what these are. This looks like a scallop dish. This seems to be a fish dish. This is seared salmon, I think, with caviar. This is another dish that looks like an appetizer. So the point I wanted to make here is that if you go to one of these really top-ranking restaurants, you see these dishes that have really tiny portions, and these are delectable. These are made with such passion. The intention is not to fill you up and fill your stomach. So the point is, if you go to one of these finest restaurants in the world, you don't go there to fill your stomach. What is it for is that you get to taste a whole range of flavors created with a lot of passion, highly creative. For example, you can see on the top left, this is Martin Bichat himself. He's the owner of the restaurant. He's one of the world's most renowned chefs. And what he does is every day morning, he just explores his own garden, which is located around the restaurant, and he picks up different things and he thinks about it, and he creatively thinks to combine these things into creating different flavors. And this is, I told you, Martin Bichat, the chef. And here you see another picture. His name is Greg Semenza. And there is a reason why I included his picture here. And I will go to the next slide and show you why. This is the inside of Martin Bichat. There are very few tables. I think there are like 6 or 7 tables, even there in 2010. And this table here to the left corner, that is where I was sitting. And to the left of me was this person. And who is he? His name is Greg Semenza, I told you. I started talking to him while we were having dinner. I was there for a conference, and we had a lot of distinguished scientists around the table with my own mentor, Eileen White. And Greg Semenza was sitting next to me. We started talking, and the moment he realized that I'm from India, he started talking with a lot of interest, because one of his postdoctoral fellows was from India, and he had a lot of stories to tell me. And we started talking about his own research. I started asking questions. Of course, he's a very famous person. I knew a little bit about his work, but it was very interesting to talk to him about his own journey. As it would turn out, Greg Semenza joined Harvard Medical School in 1974. And while he was an undergrad student over there, there was one of his friend's sons who was diagnosed with a special disease called Down syndrome, as you know, is a problem in chromosome 21. That was a beginning of a time when a lot of interest was going into understanding genetic diseases. In 1974, right after his program, he joined UPenn in the United States. And that is where thalassemia, another genetic condition, was being studied. And naturally, he was very interested in studying thalassemia. But as luck would have it, I would say, his mentor moved from UPenn in 1986 to Johns Hopkins. And he also went with his mentor to Johns Hopkins as a postdoctoral fellow. And his mentor was studying hemophilia, that's another genetic disease, and a factor 8, which plays a very important role in hemophilia. But then when he started his work in Johns Hopkins, he wanted to get a clone of factor 8 in order to continue his studies. So he contacted one of the companies and requested for this. And what they did was they sent factor 8 to him, but in addition to that, they sent him something else as a gift. This is erythropoietin. This is another very important gene, as you know. So they sent him a clone of EPO, saying that this would be a very interesting gene to study as well. So out of just curiosity, he started working on EPO. This is Johns Hopkins back in 1986. And one of the first things he did was he put this gene and he introduced this gene into mice. How do we introduce a gene into mice? You can play around with the genome of the animals in very interesting ways. You could introduce a gene, so all of the cells in the animal expresses that gene, so you can study the phenotype. You could also knock out a gene in the animal. So either you can introduce a gene or you can remove a gene completely. So one of the first things he did was he introduced the erythropoietin gene into mice. And that is called a transgenic mice, like I said. And what erythropoietin does is it plays a big role in blood development. It makes RBCs. So this transgenic mice obviously expressed the EPO and it started making RBCs. So what does that mean? Now we know that a human gene can be expressed in mice, and that was not the first time it was being done. But it still was an achievement that a human gene could be expressed in an animal and that the animal would show the functional phenotype. So the animal expressed the gene and RBCs were made in high numbers in this particular animal, especially in hypoxia. And then one of the key interesting factors was that at that point in time, nobody knew what regulated the EPO. So Greg Semenza was interested in finding how this gene was regulated, and he started working with one of his students in the lab. And as luck would have it, this was evading. Despite months and years of continuous work, they were not able to identify the factor that regulated the expression of EPO. But then one day, while he was going through the lab notebook of the postdoctoral fellow, he noticed a mysterious band that turned out to be a protein called HIF1 alpha. And HIF1 alpha is a very special protein. It induces EPO. Under hypoxia, it induces VEGF. VEGF stands for vascular endothelial growth factor. And that is an important gene that plays the role in the formation of blood vessels. So in your body, there are continuous formation of blood vessels in different locations. For example, due to ischemia, when there is a drop in the oxygen supply in our heart, VEGF plays a role in creating new blood vessels in that region. That's a good thing. And similarly in tumors, for example, deep-seated tumors, big tumors, when there is a reduction in oxygen depletion into the center regions of the tumor, VEGF plays an important role in building new blood vessels into the tumor so the tumor can get oxygen. And one of the things he did with this was he knocked out HIF1 alpha from mice. And like I mentioned before, you can either introduce a gene or remove a gene. Removing a gene is called knocking out, and he removed the gene. And I'll tell you, some of you may be wondering, how do we play around with the genome? Like cutting some part out and introducing something else in. We'll take a brief look at that in a subsequent slide. So he tried to remove HIF1 alpha from this mouse and created a knockout mouse. And one of the things that he noticed was the embryo was not viable. If you introduce a lethality in the genome, what happens is that the embryo dies in utero. So in this particular case, on gestation day 8, the animal died. And that was an interesting thing. So when they studied the dead animal, they found that there were significant problems with the development of the heart, the blood, and heart and the blood vessels and the blood tissue itself. And there was an extremely low expression of VEGF, which makes sense because VEGF plays a role in the formation of blood vessels. So the malformed development of heart, blood, and these vessels was characteristic of the knockout mice that was, deleted for HIF1. So what that meant was HIF1 is an important protein that plays a role before the birth, not only before birth, but also after birth. Before birth, it plays an important role in the development of cardiovascular system. and oxygen sensing mechanisms while the animal is in utero. But then once the animal is born, as you know, a HIF1 alpha induces the expression of EPO and also VEGF, and thereby supporting the blood circulation and the oxygen distribution. And importantly, so what this tells you is that HIF1 plays a very critical role, and also you may appreciate that the control of such an important protein becomes very important. For example, when to turn it on and when to turn it off. So Greg Semenza was able to understand how EPO was regulated and some very important aspects of the regulation of HIF1 alpha itself. But then it was not clear what turned the HIF1 alpha on or off under the right physiological conditions. For example, if the oxygen level is normal, you don't want HIF1 alpha. You need it when the oxygen level is low, because that is when you need all of these things to bring the oxygen level back to normal. And the break on the system as to how this is regulated came from a different lab. This is Bill Kalin and Peter Ratcliffe. Bill Kalin from Harvard and Peter Ratcliffe from Oxford. So these two scientists then showed that there is a very tight regulation of HIF1 alpha when oxygen levels are normal. Because of oxygen through prolyl hydroxylases, what happens is HIF1 alpha protein is targeted for degradation, and then it is quickly degraded through the ubiquitin mediated pathway. On the other hand, when the oxygen level becomes low, then the prolyl hydroxylases don't work, and the VHL mediated destruction of this protein doesn't happen, and then what happens is the HIF1 alpha is now free to do its function. So we have seen how that is regulated, and until then, everything was clean. We now understand how this is regulated. It is, oxygen controlled mechanism. Depending on the levels of the oxygen, HIF1 alpha is regulated. And if HIF1 alpha if the oxygen level is normal, then you can see high levels of HIF1 alpha that is expected. However, here is the problem. The problem is that HIF1 alpha is more than that. HIF1 alpha's regulation was not just due to the levels of oxygen. For example, in breast cancers, you see HIF1 alpha very high. And initially, people didn't understand why HIF1 alpha was being expressed even when the oxygen is normal. And then, once again, Greg's lab was able to show a connection between an oncogene, ERB2HER2/neu, upregulates HIF1 alpha. This is a genetic modulation, even under the normal oxygen, normal oxygen conditions. So what is the problem, you may ask? The problem is that we have this protein when we don't need it. And that's a problem, because think about it, what does HIF1 do? HIF1 induces the formation of blood vessels, and that's a bad thing for cancer. Because, when cancer grows beyond a certain size, oxygen depletes in the middle, and then if the tumor finds a way to build new blood vessels into the tumor, then what happens is, it has nutrients and oxygen coming in, and the cancer, it becomes unstoppable, basically. So this oncogene plays an important role in controlling HIF1 alpha indirectly, and also directly by other mechanisms directly feed cancer formation. So what did we learn so far about HIF1 alpha? HIF1 alpha, now we know, is induced by multiple mechanisms. It can be genetic alterations like HER2/neu, and it can also be due to low oxygen conditions. But the problem is that when HIF1 alpha is like a switch that cannot be turned off, this break doesn't work anymore, and then it's always on. And those constitutive expression in cancers lead to a lot of problems. And we now know that about 70% of cancers have HIF1 alpha, and this probably is one of the most important common factor between all of the cancers that we know of. And it also so happens that the cancers that metastasize, that means spreading into different areas of the body, usually have HIF1 alpha highly active. So I have shown you so far a molecule, ways in which that molecule can impact the overall health of the individual, right? So we now know the problem. And, I want to, this is Charles Kettering, and one of the quotes by Charles Kettering is something that I really like. Charles Kettering is one of the personalities that you should know, not because he has made a lot of inventions that we use on a regular basis, like a refrigerator and things like that. He's also known for creating the Memorial Sloan Kettering Cancer Center in New York. But I like him for the reason that he said something really, really meaningful. He said that a problem well stated is a problem half solved. And it's really true, because if you have an idea of what the problem is, you can think of ways and means to solve it. So now we know that most of the cancers are HIF1 alpha, and that HIF1 alpha expression is a problem in terms of regulation of controlling cancer. And the question really is, if you really want to treat cancer, if you want to eliminate cancer, what do? It's quite intuitive that you would want to block these two signals. This is a signal that drives cancer. This is a signal that drives cancer. And therefore, if you want to control breast cancer, that is precisely what you would do. You find a way to block this protein, you find a way to block this protein, and that you'd expect to reduce the cancer growth, because these are two major pathways that play a very important role in cancer formation. And that is what we have done. Daiichi Sankyo and Roche came up with these two medicines that I would say changed the landscape of cancer treatment. First, in 1998, Roche came up with Herceptin. There's a trastuzumab. It's a monoclonal antibody that targeted this protein, and that changed the landscape, changed the way we treat cancer. That's an epoch-making discovery. I would say one of the most important in the history of all cancer research. And it was highly successful. Thousands and thousands of people have found cure in Herceptin. It had, some resistance evolving eventually. It wasn't for all of the subjects. There were some exclusion criteria. And eventually in 2019, a modified version of trastuzumab, this is a antibody drug conjugate called Enhertu, by Daiichi Sankyo and AstraZeneca, was developed. But important to mention that these two drugs were developed based on this concept that I just described to you, that Greg Semenza and the two others that I showed you were able to show. And for this reason, Greg Semenza was the winner of Nobel Prize for Medicine and Physiology in 2019, along with Bill Kaelin and Peter Ratcliffe. So in 2010, I didn't realize that I was sitting next to a person who was going to win a Nobel Prize in medicine in another 9 years. But Greg continues to be a collaborator of my mentor's lab, and we have a lot of scientific interaction even now. So why would I be particularly interested in Greg Semenza? Here is the reason. Because when these discoveries were being made in Greg Semenza's lab in 1998, I was working on my PhD thesis, and I was also working on this protein not directly, but because my topic was hypoxia and cancer therapy, HIF1 was an important protein for me as well. So in 1998, as I was writing my PhD thesis, I was coming across his papers, and this is a name that I came across very, very frequently, Greg Semenza. So, once he got the Nobel Prize, I went back to my PhD thesis and took a picture of this page number 18, and I still remember the days when I was sitting and writing all this. So, summary of the first few slides without taking a lot of time. HIF1 is normally not present when the oxygen is okay, but when the oxygen is low, there are mechanisms that increase the hypoxia, and then the hypoxia induces the expression of HIF1 alpha. And HIF1 alpha is a bad thing for cancer. So if you want to cure cancer, you want to block HIF1 alpha directly or indirectly. And I mentioned two of these medicines, Enhertu and Herceptin. This is an antibody drug conjugate. And digoxin is something that I'll come to. And this is a medicine that came out of Greg Semenza's research. When they screened more than 3,000 compounds, digoxin was one of the ones that was a very highly potent inhibitor of HIF1 alpha. So I want to switch gears now. In the beginning of the talk, I briefly mentioned that you can introduce a gene in a mouse. You can change the genome in a way. You can sort of edit the genome of an animal by deleting something as in here, or introducing another gene into the body of the animal. And here I just want to talk about some new ways of doing this, because this is another fascinating story. One of the most important tools that we use in cancer in any biological research. So the story starts with two women. The first one's name is Jennifer Doudna. Jennifer Doudna is in UC Berkeley, and Emmanuel Charpentier. Emmanuel Charpentier is in Berlin, Germany, a French scientist and American scientist. And they started looking at ways in which you could sort of edit, I want you to imagine like you're typing a Word document, and you notice that you mistyped one word, or you made a mistake. You could just use an editing software, and you can sort of cut and remove what you want, what you don't want, and paste something that you want. So Jennifer and Emmanuel Charpentier, they were interested in doing that for the genome. So I think they started working in the mid 2000s, and they have developed a beautiful system that is based on a bacterial system called CRISPR-Cas, which I will come to in a minute. But I just want to show you what they were able to achieve by introducing and deleting genes and introducing tiny, precise mutations. In the first picture, what you're seeing is black mice on the left. And what they have done, they were able to very precisely change one gene, introduce a tiny mutation in one of the genes, and they were able to convert the black mice into white. So that is, that makes us think that, what a tiny mutation in one gene can do. What does it even mean to be black or white? That's a profoundly philosophical question, and an important application of this technique. So likewise, some kinds of snails, you have the shell of the snail in a certain orientation. It's coiled either in the right angle or left angled orientation, and that is controlled by a gene. So Jennifer and Emmanuel Charpentier, they were able to use this technique to change one tiny part in the genome, and they were able to change the orientation. So, now the coiling direction completely changes as a result of this change in the genome. Similarly, there are applications in agriculture. You could change the genome of a tomato, and you could keep the tomato same in every possible way, but at the same time, you could make tomato plants that produce tens of thousands of tomatoes more than what you would otherwise. So significant improvement in the yield of the tomatoes by just simply controlling the genome. And that is just, as the schematic shows here, this is basically using a pair of scissors, going in and cutting the DNA. And you probably, and this audience, I think you are familiar with the sizes of these things that I'm talking about, nanometer kind of dimensions. So you can imagine that, taking a DNA out of the cell and cutting it is not practical. So we have to do something else, right? How do we cut the genome that is within a chromosome in an organism? So, Jennifer and Emmanuel noticed something that is very fascinating that's happening in bacterium. So this is interesting because, now we are all talking about viral infections and ways of fighting the virus and our immune system fighting the virus and things like that. But here I want to tell you the story of just a simple tiny bacterium. And you may be surprised to know that a bacterium has an immune system that is powerful enough to keep viruses away. And the system works like this. The system is called Cas system, Cas1 and Cas2 complexes. So this is a virus, and this is the bacterial cell. And when a virus attacks the bacterium, what it does is it inserts its genome into the bacterial cell. So this is the viral DNA. As it comes in, what the bacterium does is it finds a way, using the Cas1 and Cas2 complexes, it goes in and cuts the DNA and cuts tiny pieces out of it. And then it brings it and it incorporates into its own genome. This is bacterial genome, and in special places, those little pieces. So, scientists have known that bacterial genome have what is called CRISPR, what stands for clustered, regularly interspaced, short palindromic repeats. So these tiny repeats are seen throughout many types of bacterium, and people didn't know why they were there. And it so turns out that what this bacteria is building is a library, a history of the kind of viruses that attacked this particular bacterium in its life. So by integrating tiny pieces of every virus that came its way, it is actually creating a memory of what once attacked the organism. And then what happens is that these tiny repeats will form a complex called a CRISPR-Cas complex. And then the next time this bacterium is infected by the same kind of virus, this complex is very quickly able to identify the unique sequence that actually is stored in its memory. And then, this is a nuclease, Cas9, and Cas9 just starts chewing this DNA away. It just binds to the right sequence and it just starts chewing it up and thereby destroying the invading viral genome. This is an interesting concept because, these two scientists, Jennifer and Emmanuel, they thought that this is very interesting. This is like, the bacterial cell system is just hiding in with a pair of scissors waiting for the virus to come in. And the moment the virus comes in, it just attacks and just cuts it off. So, and that's a very brilliant concept, which, sounded like in the way we edit, like I said, the documents we type. And, I just want to want you to think about a specific example of correcting mistakes or correcting problems along long cables. In high-rise cables, electricians have to somehow find where the problem is, and that's a very difficult process. They have to, somehow travel along this along this electric line, get to the point where there is a problem, and then they have to address that. So I just want to play this video that kind of, shows you how this is done by humans and then we will come to what Jennifer and Emmanuel was able to do. I want you to be really scared seeing this. Did you see that? So that's how we fix, problems along long cables that is so high in the sky. It's so difficult to get to the point, get to the precise point where the problem is, and cut it and splice it and change fix the problem. So Jennifer and Emmanuel, looking at the way bacterium protects itself, got an idea, a beautiful idea, and they thought, why don't we, hijack this bacterial system, engineered it to make it a programmable kind of genetic scissor, like a pair of scissors. Why don't we make it like a programmable genetic scissors? And that is precisely what they have done. And in the next video, I want you to watch this video, and I'll display in a moment, and you will see the guide RNA, the sequence that guides this whole complex to the right sequences. And this is the Cas9 part, which is the nuclease that I showed you. And in this movie, you will see how the complex moves along the DNA and looks for this is the Cas9 portion, and that is shown in blue, and then the guide RNA, like I showed you, that is in the protein with the sequence. So now remember, the guide RNA is the one that has that sequence, that specific sequence. So when this complex is in the cell, if you could deliver it properly, then this complex will move along the DNA, and it will look for the sequence that actually perfectly matches with the memory of the bacterium. This is exactly how the CRISPR-Cas9 system works in a bacterium. So, and then, like I told you, this is a nuclease, and let me play that part again. The nuclease, which is Cas9, sort of very precisely cleaves the DNA at that point, and you can really engineer that to precisely define. So, and then what cell does is that this is DNA repair machinery. So once the DNA is broken, the DNA repair machinery comes in and tries to fill in. And this is actually a very highly error-prone process. So this is how mutations happen. And let me pause here because something very important is happening here. So suppose you introduce a mutation here, and let's say this part of the DNA is a gene that you wanted to sort of silence. You can sort of silence the whole thing by just introducing a particular mutation over there. So what happens is, you can introduce a stop codon, and then the remaining part of the DNA is never read, and you can sort of turn off the expression of that gene. So this is how the system can be used to very precisely engineer and direct to a specific sequence of the DNA and turn on and turn off the machinery. And this video is again showing you how precisely you can control them. And you could do much more than you just snipping it. You could change the codons. You could introduce the stop codon, and also you are able to bring in other factors that would prevent the expression of this gene that you would see in a few minutes. So using this CRISPR-Cas9 molecule, you're able to bring the regulators of gene expression and sort of control the expression of genes that you don't want to express. And these are just showing you different ways of it doing that, different factors being brought in to sort of control, to sort of prevent the RNA polymerase from transcribing the rest of the DNA. So you could do that for improving the expression, like in this particular case. This is DNA polymerase that is inducing the expression of the genes. And then this is an example where you could stop the expression of the genes by bringing in other factors. So you are changing the structure of the DNA either by introducing mutations or by bringing in other factors that interfere with the expression of the genes, and you are able to sort of control the whole thing. So this molecular scissors idea was developed by these two scientists. And here what you just saw was that you could use this to paint chromosomes. You could, for example, add a fluorescent protein here, like GFP, green fluorescent protein as shown in here. So you can combine that with this factor, and you could sort of paint the whole chromosome if you wish. And that tells you very important information about the disease pathology in some cases. So that is what CRISPR-Cas9 system that was developed by Jennifer and Emmanuel. So that is another example of a beautiful tool that is applied to control the genome of the cell, to sort of precisely manipulate the expression of the gene, like what the Greg Semenza lab was able to do in the beginning of the talk. Now, where does this go in future? If you can manipulate the gene, and it's already there, you can manipulate the gene of a fly to introduce a wing, to remove a wing. You could remove one eye. You could create a fly with 3 eyes, 4 eyes. You can change the color of the eye, you know. And the examples I showed you, if all of that is possible, this can be applied in humans as well. If, for example, if you want to, this is designer babies, and it's not scientific fiction anymore. It's not science fiction. This is a reality, in theory at least. And that brings in a lot of ethical questions, and I'm not talking about the ethical aspects of it, and we should probably never go there. But the point is that you are able to use these molecular tools in order to change the genome, and you could create organisms and even human beings with the designer phenotype that you would like. For example, you want babies that are very high in IQ, you want no baldness, you want perfect vision, you want to reduce the risk of certain diseases, you can have the voice control of the person, you can make the person a sprinter by increasing the physical stamina. All of this is possible with even today's technology. Of course, there are improvements needed in the precision, in making sure that you don't do anything unnecessarily complicated. You don't want to accidentally introduce something else that you don't want to do. So, I mean, the technology is still being perfected, but this is an important tool that has revolutionized the biological and medical research. And for this discovery, Jennifer and Emmanuel Charpentier were the winners of Nobel Prize for Chemistry this year. And this prize was announced just a few weeks ago. And now you understand what they did in making some of these very complicated processes really simple. So I want to switch gears again, and we talked about genes and introducing genes, removing genes, manipulating genes. We looked at the ways in which it can be done in the cell by using tiny molecular scissors. Now I would like to take you right into the cell. I want to ask you this question: have you seen cells in action? If you took your cell out and if you put it in a plate, in a petri dish, and you give them right nutrients, and if you look under the microscope, this is how they will look. You are looking at your cells. And then if you build a special system like I did about 20 years ago, when I was in Eileen White's lab, I basically designed a system, a microscopic system where you are able to grow these cells in a petri dish, and you can grow them and watch them. You can make movies of these cells dividing and dying for long periods of time, maybe days into weeks. So I just want to show you the system that I made, and there were a lot of interesting experiences that I've had while I was working with that, making movies. We have had distinguished visitors coming to see this, and one of the distinguished visitors that came to my lab, I think that is in the next slide. I will come to that, but before that, let me show you how the cells move. This is the video playing, and as you can see, these cells are slowly moving now. This is a time lapse, of course. It's not a continuous movie. This is a fast-forwarded movie, but you can see the cells dividing into two. You can see the cells increasing in number. There were a few in the beginning, and now you see many more, and that is because the cells are absorbing these nutrients from the medium, and they are dividing and multiplying. This is a fascinating thing that I was very excited to be a part of. And one day I was doing this experiment, and I got a notification that a distinguished visitor wanted to see this facility. Excuse me. Bear with me as I have a little bit of weather-related allergy issues. So I pause in between, and just please bear with me. So one of the visitors, the distinguished visitors that came to my lab, who wanted to see the movie making, wanted to also know a little bit about the scientific research and how do you run a lab and how do you study viruses. And I showed him this setup. I showed him that, this is the setup. You're looking at the movie making facility that we created. This is called computerized video time-lapse microscopy. You can see this, you can put the cells in here, and you can follow these cells for many generations, and you can actually do much more than that, which I will show you in a subsequent slide. But let me show you the celebrity who wanted to see this facility. Some of you may remember, this is a famous Indian actor, Kamal Haasan. And this was the time when he was making the movie Dashavataram, and in that he is playing the role of a scientist. And he visited some of the major US labs, and ours was one of them, and he came to know about my facility, and I was very happy to spend some time with him. So here is a set of movies that when I play, you will see how sometimes the cancer cells, this is a cancer cell on the left side, cancer cells play a trick on us. When I say play a trick on us, what I mean is that sometimes they pretend that they're dead, but then they're not dead, and they wait for the right time for them to come back. When I play this movie, you will see on the left side, oops, the cells dying slowly, and that's what I thought too. On a Friday evening when I set this up, I thought these cells were dying, and then I came back and I saw them dead. And like I said, the computerized video time-lapse microscopy allows you to sort of, put some medium back, meaning, you put some food back to them to test to see if they are really dead. And that's what I did on a Monday morning. When I came back, what I did was I gave them a little more food. And what happened was these cancer cells were they were not really dead. They were crawling out of their shells, and they were able to take up food, and they were able to at least come back to life for some time. So I want to switch gears again and bring to your attention the first example I gave you of HIF1 alpha. And now I want to talk about how these principles help us in treating cancer, right? Some of you may want to know how we discover medicines. So you're looking at 4 movies. This is the same kind of cells in all of the movies. In the first movie, it's just the cells and food, and there is nothing else. And sometimes if this is a cancer cell, and if the cancer cells are fed by 2 different pathways, if you remember, the HER2/neu is one of them, HIF1 alpha is one of them. There could be different pathways. And if you block only one of the pathways, nothing happens. Why? Because the cancer cell can still survive using this alternate pathway. If you really want to kill them, you want to target both pathways and block both of them. So in this movie, when I play these, you will see that in this, the cells are just normal. And here, you are just blocking one of the pathways, the left one. And you will see that the cells don't care. They just sit there and laugh at us because it has the other pathway that is still active. In this movie, you will see the other pathway blocked. And once again, you will see that the cancer cells don't care. And you will really see something very interesting over here, and that is blocking pathways 1 and 2 together at the same time. And watch, this is not blocking any pathway. This is blocking one of the pathways. This is blocking the other pathway. This is blocking the pathways together. So this is sometimes called synthetic lethality. So you sort of, target 2 different things in the cell, and together they kill cancer cells. So I gave you, by now, you should have an idea, a fairly good idea of, some of how we identify some of the cellular targets to target certain diseases, and how we design tools to sort of precisely make those changes in creating some of the model animals, sometimes even directly going in and changing the gene, which is the gene therapy proposition that is very attractive in cancer therapy. Gene therapy is still being refined, but using the technologies that I showed you, there is a great promise that we'll be able to change the gene, take out the mutated gene, put the gene back, the right kind of the gene back, and you could hope that you would be able to treat cancer. So this is an experiment that I actually did with collaborating with Bill Kalin's lab. So now let me switch gears and come to the drug discovery. We are, I think more than halfway through, and really tell you some very important concern that people have. We live in at times when people are excessively concerned about consuming any chemicals. They don't want chemicals, and they would rather go for natural alternatives. They think the natural means not a chemical. There is a public perception that if something is natural, it's not a chemical. So let's look at what is the difference between a natural and a synthetic. For most of the audience here who are, high-level scientific students, this is probably not that important because you already know, but I just would like to still give you an idea. So in this slide, you're looking at a lot of different chemicals, right? And I have a question. How many of these molecules actually come from plants? Is it none of these? Is it all of these? Is it maybe 25%, 50%, 75%? One of the things I wanted to also do along with this webinar today was to sort of use some kind of an online poll. And online polling was not possible with Gmeet, so I was not able to do that. I had a different plan, but that software had some issues, so I decided not to do that. If I was doing the seminar live, then I would be able to see you raising hands, but I still would like you to just pick one answer, A, B, C, D, or E. And let me give you a few seconds for you to think. And I really want to know how many of you got the correct answer. So the right answer is that all of these molecules come from plants, and they are implicated in different diseases, as you can see, including cancer. So this is paclitaxel, as you know, is a very important cancer drug, one of the very important ones. In this slide, you see some plants, right? This is foxglove, willow, mayapple, and poppy. And do you know how many medicines, do you know how these plants play an important role in drug discovery? Let's take a look. Digoxin, the medicine I showed you in one of the earlier slides, is actually a product of this plant. There's aspirin, the wonder drug, that comes from willow. Morphine comes from this plant, and etoposide, which is another cancer drug, it comes from the mayapple plant. So, the truth is that everything in this universe is chemical in some form or fashion, right? And plants still, nature is one of the most important sources of chemicals. So, and that's where we would normally start. When you want to develop a drug, you look into nature, because there's a very vast body of historic texts, Ayurveda, for example, Chinese traditional medicine. So from these historical texts, we have an idea about what might work. That's only a good, a reason good to believe kind of a suggestion, but that it's not confirmed yet. But once you have an indication, then what you do is you do a lot of things, to gather information about the kind of plant that is, and therefore the kind of molecules that it may have. If you don't know, you start extracting it. You start doing some crude extract bioassays. This is just basically for those who don't, have an experience in this. It's basically cells grown in a plastic petri dish. And then you could just directly add some of these extracts into it, and you see if something is happening. If something is happening, then that means it has some activity. You can do different versions of that by fractionating it, purifying it, and then maybe even identifying the active compound in that, because a typical drug extract may have tens of thousands of compounds. And sometimes one activity may be inactivated by something else. So you may not find anything in the crude extract, but you may be able to isolate the active principle and be able to identify something very valuable. And then, you could take this molecule and you could further modify it. There's something called QSAR. It stands for quantitative structure activity relationship. What it is that it sort of correlates the function of the molecule with various functional groups that the molecule has, and then modification of those functional groups to modify the final activity of the molecule itself. So you can really do that using the quantitative structure activity relationships. And then, before it comes to humans, of course, you have to go through a lot of hurdles, like you have to know the toxicity, so the toxicology, pharmacology, absorption, distribution, metabolism, and elimination, and PKPD, the pharmacokinetics and pharmacodynamics. I won't go into the details of it, but if you want to know more about it, you definitely can ask me questions. And then final formulation into an oral or a topical or a parenteral, different kinds of formulations that we can use as a medicine that eventually will go to the clinical trials. And then if it is successful, then we take it to humans. So, as chemists and biologists, you will be interested in the processes that are important. I just mentioned about the plant extracts. And sometimes you need to separate out the individual actives from these plant extracts. And the separation, as you know, is done by chromatographic methods. And then, the structure determination usually is done by crystallographic and spectroscopic methods. So from plant extracts into pure compounds that are isolated, the activity tested in biological assays, and now we know what the structures are thanks to the spectroscopic methods that has been developed. So this is the Taxol drug that I was just talking about. Taxol is one of the most important medicines out there. It saved millions of lives all over the world. It is still being used as a cancer therapy. And it comes from this plant called Pacific yew. And, I just want to give you a flavor of the structure-activity relationships, bringing in some chemistry, because, unlike the previous webinar that I was giving somewhere else, which was mostly chemistry-focused, I just want to bring in some biology, because there's a biochemistry, molecular biology department. So you have seen some of that. But then I just want to bring back a little bit of chemistry. and really show you. This is really, really exciting because now you know the structure of a molecule, and you can sort of tweak the structure and sort of improve the function of the molecule. So this is the molecule Taxol, and Taxol is not one of the simplest compounds. This is an extremely complicated structure. Of course, you know that these molecules are three-dimensional, and there are so many different functional groups, and many rings in there, and not all of these functional groups are very important as far as the function of this is concerned. Some of you may know that Taxol is a microtubule binding drug. It goes into the cell and it binds to the microtubules and it stops the cell division and it kills the cell, and that's how it functions as a cancer medicine. Now let's look at what functional groups are really important and what can you actually do with this. So if you look at this, the N-acyl group that is shown here is very important here as far as the function is concerned. And if you replace this group with a methyl group, you immediately reduce the function almost 19-fold. So you can see this is an example of how a small part of the molecule plays such a big role. And the C3 bound nitrogen it can be replaced by oxygen atom without any loss of activity. So that's another important thing. Sometimes you may be able to remove something without losing anything. And C3 amide acyl group is critical, but it can be alkyl in nature. It can be aromatic or alkyl in nature. And then, here, if you remove this hydroxyl group, if you introduce a different group, what will happen? You lose the complete activity of Taxol. Then if that simple change over here eliminates its microtubule binding capacity for whatever reasons, and it still be cytotoxic, but it will not be microtubule binding. And that is not a good news for cancer therapy because we depend on microtubule binding. If you really want to know why, I'll be happy to answer that question. But in cancer therapy, the function is lost if you change that functional group. So we have seen that the molecule. So what did we see so far? In the beginning of the talk, we saw how we identify a molecule as a key reason, a key factor in creating a disease. And then we saw that there are molecular tools that we can use to sort of create changes in the genome to sometimes address those changes and modulate those pathways. We've also seen that a lot of compounds come from plants, and we have seen the steps through which we can isolate and purify and sort of functionally characterize, even structurally characterize some of those compounds. We have seen that the structures are complex and that the functional groups play very specific roles, and you can play around with some of these functional groups and change the function of the molecule, and that's how we design more and more powerful medications. Now I want to show you three really exciting examples. Acetaminophen is a widely used pain medication called Tylenol, and then quantitative structure activity optimizations are possible on this molecule, and by doing so you can, and as you know, too much of Tylenol is not good. This is basically paracetamol that is present that's in the Indian market, and one of the problems that we face is acetaminophen overdose. If somebody takes too much of this drug, because it is metabolized in the liver, it induces acute liver toxicity, and the person can die immediately. So this is also used as a suicidal agent. People take a lot of times. There are a lot of cases where people use acetaminophen for committing suicide, and that happens because of the hepatotoxicity of Tylenol or acetaminophen. You can actually make slight modifications in the structure of this molecule, and that will eliminate the liver toxicity. And there are a lot of research happening in this field as we speak. Here is another example of introducing an oxetane ring. A simple introduction, I shouldn't say simple, but just a simple one-step structural change can drive the metabolism completely different. it can drive the metabolism away from this cytochrome P450 enzyme that usually metabolizes this molecule into a totally different pathway that reduces the drug-drug interactions. And this is a fascinating thing. This is, in isomerism in chemistry, you probably have seen and studied that sometimes the molecules exist in different stereoisomeric forms. And when two rings are connected by a single bond, they can freely rotate most of the time, as you see on the left side of the figure. But then when you introduce sometimes bigger groups, then what happens is what is called steric hindrance. And because of the sheer size and the repulsion and the steric interaction between these functional groups, the rotation will be inhibited, and the bigger the functional group, the more inhibited the rotation will be. So, and this situation is very valuable. We can exploit the situation in drug design. This is called isomeric forms. Heteroisomerism is actually good. For example, in this figure, you are looking at a protein called NAV1.7. NAV1.7 is a gene that is extremely important in sensing pain. If you lose this gene, or if you have loss of function mutations in the gene, you know what will happen? What will happen is that you will become insensitive to pain. There is a disease condition called congenital insensitivity to pain, and those people can never feel any pain. So, and that's an extremely dangerous situation. So people who are suffering from congenital insensitivity to pain usually die very early because, they can put their hand right into fire and they won't know it. They could be burning and they won't know it. Such extreme is the phenotype of that disease. So NAV1.7 is a gene that we could manipulate. And in fact, that is done by using some heteroisomers of certain kinds of molecules, in this particular case, quinolone sulfonamide derivatives that are sterically hindered in rotation. And these are effective blockers of NAV1.7. So if you introduce this kind of an isomerism in this molecule, you can generate a molecule that now blocks NAV1.7 and you block the pain. So, that's an attractive proposition in designing painkillers. So these are some of the ways in which you can sort of, play around with the structure of the molecule and then, have an impact in a specific disease condition. Now, let's take a look at what does the drug development entail. You may be asking, okay, this is all fantastic ideas, and how much does it cost and how long does it take? Let's take a look at that. This graph is showing you, how much does it cost. Of course, it depends on what area, and all of the areas, all of the different types of diseases are not the same. On the left, you have oncology, that's cancer, which is probably the most expensive drug development, and that is, as you can see, very close to $4.5 billion. This is, billion with a B. So $4.5 to $5 billion, and in the rest of the conditions, not too much different. In all of these cases, we are talking about at least $1 billion. Now, how long does it take? And when I go through this slide, I just want to give you a general idea of the various steps that you have to go through in developing a drug and release it into humans ultimately. So first, you understand the disease pathology, and remember the HIF1 alpha story. Molecular mechanisms, you need to understand why the disease happens. Structural chemistry and screening, remember the planned extractions and the spectroscopy I told you, and the structure-activity relationships. And the example of Taxol, where we modified the subgroups to change the function of the molecule. And also remember the different ways in which you could introduce isomerism and sort of develop new drugs. We talked about target identification. Think about the movie making machine and the ways in which you could see if a particular agent is killing the cells. Synthesis and purification, we talked about it. So we have to synthesize and purify it in large quantities. First, you look at the efficacy in vitro systems. Then you look at it in animals. It's called a preclinical phase. And once you pass all of these stages, and this takes, sometimes 10 years, typically, 6 to 8 years, very, very long time, and millions of dollars in expense. And here, how much money it costs is something that you can't even calculate. And then, you take into first in humans. We start with phase 1. In phase 1, then phase 2, and then phase 3. In phase 1, we look at the safety of the molecules. And in phase 2, you also look at the safety and efficacy. And in phase 3, usually in a very large population, you get the real evidence for the efficacy of the molecule. And there are various regulatory approvals required for this, and I'll come to that. But in the United States, anything to put in humans, you need approval from the US FDA, and that is usually through an IND application. An IND application is an extremely complicated and expensive process. The application fee, I'm working on that as we speak. So I know a simple IND application alone can cost anywhere from $2.5 to $3 million, just the application fee. And then the expense of conducting the clinical trial is not even counted there. And that phase 1 through phase 3, it usually takes 1 to 2 years. And then you apply for what is called NDA, New Drug Application in the United States. In Europe, it's different. In every country, it's different. So you apply for that. And once you get the approval, the drug is marketed and it's released into the market. But once you release into the market, that doesn't mean you stop. You can continue to study. You should, in fact, continue to study the drug to really see if it is causing any kind of problems in humans, any kind of toxicity. Is somebody dying? Is there any unwanted side effect happening? So you continuously monitor, which is an extremely expensive business to do. And you can see, for many years, and it can take anywhere from $1 to $5 billion. So we are actually talking about billions of dollars in total cost in bringing a drug to market. And how long does it take, and what are the chances? Before that, let me show you what are the agencies. These are the information that is not very easy to come by. In the United States, it's called FDA, as you know. In Europe, it's EMA, European Medicines Agency. In China, it's called State FDA. In India, we call it Central Drug Standards Control Organization, or also known as Drugs Controller General of India. In Australia, it's called TGA, and in Canada, it's called Health Canada. So if a company is developing a drug, depending on which country it is trying to market, it will have to apply to that regulatory agencies and do studies that satisfy those agencies in order to get approval. Now, what percentage of the drugs that we start with phase 1 actually get to approval? You can see that it's extremely low in case of cancer, which is why it's one of the most expensive drug development programs. Only 3% of what goes into phase 1 eventually get approved. And even the best case scenario, it's no more than 1/4. So you can kind of get an idea that majority of more than 75% of the molecules that we start with in clinical trials don't make it all the way to the end. Now, I briefly mentioned, and we are towards the end of the presentation, I briefly mentioned that a drug, despite showing safety and efficacy in all of the phase 1 through 3 trials, it can still have problems. If you take these examples of greatest failures, and probably Vioxx, marketed by Merck, is one of the most tragic cases of failure of the drug. It was withdrawn after being 5 years in the market. It costed the company, the recall exercise alone costed like $6 billion. It had adverse events in so many different people, hundreds of thousands of people affected by this drug Vioxx, and the problem was cardiovascular problems. And then another example is Pfizer's Bextra, which is also one of the worst in the history. It also it was a medicine for arthritis, but it caused heart attacks and stroke in some people, and it costed Pfizer a large amount of money. It's one of the largest criminal fines imposed on any US company in the history, because $1.2 billion was fine alone. And Baycol, that is another case by Bayer. Again, this was recalled because of people dying, hundreds of thousands of people dying due to muscle wastage and complications related to that. So these are three examples of what can go wrong despite all of these very carefully fine-tuned exercises. Now, let me come to the end of the talk. Here is we began, Martin Mishat in Scotland. And you see that the room where I was sitting with Grit Semenza. And we know that, this is a delectable experience, and some of these finest places, the dishes are never meant to fill you up. What is it meant for? Like I mentioned in the very beginning, it is meant to give you a flavor. It is just meant to be an appetizer, so that you taste it and you try more and more, that you become hungry. You become hungry, and that's the opposite of what we expect from a restaurant normally. You normally go to a restaurant to stop hunger and don't want to be hungry. But in these finest restaurants, you will be hungry because that's how small they give you, and that's how the flavors are manipulated. It's made with a lot of passion. And this presentation was made the same way. It was made with a lot of passion. It was not meant to give you everything you wanted. It was just meant to give you just a flavor of different things, just meant to be appetizers, so that at the end of listening to this talk, you become hungry. You become hungry for more knowledge on how research helps people and make lives different. And that is what is intended. And if I achieve that, I would consider this a worthy exercise. With that, I would like to end here. Once again, I would like to thank you all for joining and giving me your precious Saturday morning, and I will be happy to take any questions.
Thank you, Dr. Robin Matthew. Now the session is open for discussion. Participants can directly ask, if you want any questions, they can directly ask to the speaker, or you can type the questions in the chat box.
I hope my voice and my throat issues didn't interfere, and you were all able to hear me properly and see my screen.
Yes.
Thank you.
Sir, I have a query.
My name is.
Anyone can unmute your mic and ask.
Proceed, gentlemen.
All right, just start speaking, and if somebody else is speaking, if you want to stop, you stop. Otherwise, just continue. Only one of them, I guess.
Okay, sir. Yeah, my query is, all of us know quite a bit about the CRISPR-Cas9 system and how it is used to. It has multiple applications, but my question is, yes, about the ethicality, which you didn't want to go into, but in my opinion, the CRISPR-Cas9 system or editing tools can be used to correct diseases if needed, but they should not interfere with um, the I know, I mean, where the application should not be where you are enhancing inequalities, is my opinion. What are your views about this, sir?
You can enhance the expression of genes as well. You can,
But
you know,
why enhance it if we were born intrinsically the way we were? I mean, I know it's a debate. So if the diseases need to be corrected, let's use the technology. But if uh, we are enhancing inequality, for example, maybe our physique or anything else that we want and have want to enhance, but that could be treated as unethical, in my opinion. Yeah, that's why I want to know your views, sir.
yeah,
So is
yeah.
it okay
So,
to enhance
yeah.
rather than just use it to correct diseases?
Yeah, yeah. So let me give you my perspective. I'll give you a couple of points. First of all, at the outset, before I even answer the question, I want to reiterate that I don't want to talk about the ethical aspects of it, because, like any technology, it will be, it's a debatable topic, and anything can be misused. If you have a knife in your hand, you can actually use it to save somebody. You can use it to kill somebody. Like that, this tool also has very powerful positive and negative impacts. So I'm not discussing the ethical parts of it. If you do want me to talk about it, we can. But the important thing is the first part of the question. There are cases where you want to legitimately improve the expression of the genes. In cases where something is silenced, in cases where something is under-regulated, in cases where the overexpression becomes beneficial and harmless. It can be in plants, it can be in animals. For example, in plants, you could develop, one of the actual examples that people have used this for is to generate, let's say, mushroom. Mushroom is an actual example. Mushroom can be genetically engineered to keep it just the same way as far as every other aspect is concerned, but you could introduce better disease fighting mechanisms. That is one example that can have revolutionary impact in agriculture and world poverty. In diseases, in some cases, you probably want the expression of certain genes enhanced. For example, if it's a tumor suppressor gene, you want to bring it up. And that kind of dialing up of the expression of the genes are actually possible using this technique. Not the original version of it, but modification of this technique.
Okay, thank you for your answer, sir. But okay, I mean, you are right when you say that some genes need to be, which are under-regulated or silenced, they need to be corrected. So I agree to that part. What I meant was that using it for just for the heck of it, just to maybe build a good physique, or you want to change the color of your eye, and those kind of things. So how ethical is it to use it for those kind of things?
Yeah, yeah,
Correcting
yeah.
the genes is
Yeah.
fine. Yeah.
Oh, yes, I totally agree with you. Again, a lot of things can be done, and many of those are going to be hopefully good. But some of that definitely can also be bad. And that is where the regulations play an important role. Hopefully, the government will play a role, more regulations will come, more ethical control will be exercised on what you can and you cannot do with a technology like this. Definitely, yes.
Thank you, sir. Thank you very much for the answer.
Any other questions?
Hello, sir. Like I have read articles where people are talking about certain natural enzymes obtained from plants or microorganisms which can prove anti-cancer. So I want to like even nanotechnology is giving us a lot of new aspects in which we can look for the anti-cancer therapies. So can you tell me like how what balance we have among these anti-cancer therapies, and is really playing with genes important? Like, is it required in spite of availability of all these other therapies?
Yeah, again, you have to understand that treating cancer is not a simple thing. Why is it not a simple thing? Because cancer is a very complex disease. Cancer is actually not one disease. In fact, it is said that cancer is like 200 different diseases, because cancer of one tissue is not necessarily the same as cancer of a different tissue. There are significant differences. So our approach should be to find whatever works. You are right that nanotechnology and applications of nanotechnology can be used. There can be approaches where, for example, hyperthermia. Hyperthermia is a standard way people have tried to cure cancer. At least in some kinds of solid tumors, hyperthermia has been shown to be working. So the question is, what do we go for? I don't think we are at a point where we can say that we don't need to target a gene, let's just use nanotechnology. We are not there yet. Of course, the hope is that some of these technologies will develop to a point where you don't have to specifically target a gene. In fact, there are problems of targeting specific genes because cancer is a very complex disease. If you block one pathway or a protein, it can find something else. So that's why targeted therapy often fail, and you get resistance over time. So if you target something that the cancer cannot live without, there are examples of that. For example, acidic environment, hypoxic conditions. There are certain conditions that drive cancer growth. So if you target those phenotypes rather than the molecular targets like individual proteins, some people argue that may be a better way of doing it. But I don't think we are in a position yet to be able to say that we don't need to target any molecules. We can just go via this approach. Does it make sense?
Yes, sir. Thank you.
Sir, I would like to know about the current status of personalized medicine.
Personalized medicine is again a very exciting new thing in the field. Personalized medicine is being practiced in many disease cases. The truth is, it's somewhat like buying ready-made clothing versus stitching your own. The second is tailor-made. So as far as health is concerned, you can look at your genetic makeup, you can look at your disease, you can look at your specific condition, and you can tailor-make the therapy to your need. And that is personalized medicine. With the complete sequencing of the human genome, we understand the human genome much better now. And with this new technology, some of the technologies that I mentioned today, we are even a step closer to delivering personalized medicine. But then again, it's one of these areas that's progressing. A long way to go before we can actually reap the full benefit of that.
Thank you, sir.
Another question from a YouTube viewer, Sangeeta Bharadwaj. She's asking that she is curious to know how to grow the cancer cells in Petri plate.
Okay, in fact, cancer cells are the only cells we can grow in petri dishes. The reason is if you take a normal cell and try to grow it, it's not easy. They won't grow. It will divide for a few times, maybe 20 to 25 times, but after that it'll die. If they don't die, it will enter into a state called senescence, which is basically an arrested state, and it'll just sit there without dividing forever. In order to make a cell grow in a petri dish, you have to sort of either make it cancerous or do some manipulations like providing additional factors and things like that, which will extend slightly, but oncogenic alterations are usually required to immortalize a normal cell. But again, coming back to your question, how do we grow cancer cells in a petri dish? You can actually, dissect a piece of the tumor in biopsies, for example. You take it out and put it in just regular sterile PBS, and then you bring it to the lab. Everything aseptic, sterile, and then in a sterile condition, you can have nutritional medium that you can buy. You put it in a plastic plate, you put the medium that has all of the necessary nutrients that the cancer cells will need, and you just chop it. You literally, this is how you do, and I've done it hundreds of times. You chop it with a pair of scissors, just cut it into small pieces, and then you put it in this plastic dish, and you incubate at 37, and you definitely add some growth factors and things like that, and they will grow. And in fact, the movie that you saw is an example. The movie that I showed was a few cells in a plastic plate fed with nutritional material. Did I answer your question?
Yeah, that is from a YouTube viewer, Dr. Matthew.
Oh, okay.
And another question is from Sanam. Okay, he says that he is currently working on a project related to.
Could you repeat it one more time? Sorry, he's currently working.
Yeah, working on a project work related to white rot fungal metabolites having antimicrobial activities, and he found some pathways and metabolites are same as plants also, or as plants also produces. The metabolites like taxols, phenols, and any others, and mushroom and white rot fungi also produces. So his question is that is the pathway, these pathways same for metabolite synthesis in plants and mushrooms?
I don't think I understand the question completely. Could you repeat the second part of it? What is the question again?
Let me ask. Sanam, are you online now, Sanam?
Yes, ma'am.
Yes. Yeah. You can ask him directly.
Yes. Thank you so much for this presentation, sir. Sir, I want to ask you that I have studied from many literatures that the compounds are same, means metabolites are same like in white rot fungi like mushroom and plants. Like, sir, some example taxols, phenols, and any other compounds like some steroids. So that question is that, sir, the pathways for biosynthesis of the metabolites are same as plant and mushrooms. I was confused in that.
Well, I'm not an expert in the field, but I don't understand why not. Why does that surprise you?
Sir, actually found the pathways are almost same. That's why I just asking you that.
What do you mean by the pathway? What pathway? I mean, there are different kinds of pathways. There's synthetic pathways, there are biological pathways. What kind of pathways are you talking about? Maybe I'm missing something here.
Like succinic acid pathway, coordinate pathways, like malonic acid pathways, like that, sir. Terpenoid biosynthesis.
Yeah. Yeah, and can you frame the question one more time?
No, sir, my question was only that.
Can you say it one more time? Sorry, I just want to make sure I understand it.
Sir, my question was the pathways are the same in plants and mushrooms, like they produce same metabolites and following the same pathways. Means I wanted to ask that question only, sir.
Yeah, I will have to say it is possible. but I have to say that I'm not an expert in that particular field. So, I would think that is possible. That's it.
Sanam, hope the answer is clear to you. I think she asked actually is the pathway same for the synthesis of taxol in plant and mushroom? I think that is a question she meant.
Plants and mushroom. I'm sorry, I don't know.
Okay.
Not my field, and I don't think I can give you an answer for that.
No problem, sir. Sir, that's fine. Thank you, sir.
Thank you for the question, though.
Okay, another question from Nupur Srivatsa. She's asking, which media do we use for growing carcinogenic cells? Can we use agar media?
No. Normally, mammalian cell culture media is different. In agar media, you use for bacterium and things like that. When you want to grow mammalian cells, what you do is you buy these mammalian cell culture medium. It all depends on what the specific type of cell needs to grow. The nutrients and growth factors are all different. In fact, there are many kinds of many different kinds of mammalian cell culture media that are present and that are available, and most of them do the same thing.
Okay, I hope that question is clear for Nupur. Next is from a YouTube viewer, Meghma Sarkar. She's asking, is there any role of stem cell or its culture in personalized medicine?
Yes. Stem cells, just the way that they function, sometimes one example I think is, cord blood, right? Stem cells are original cells that are assumed to be before the transformation that happened in a cancer cell, for example. So certain types of stem cells can be, if you have it collected from the same person stored away, those stem cells can be used to treat the same person in certain types of cancers. Not all types of cancers, but in some types of cancers. In certain types of blood cancers, for example, if you have the bone marrow from the same person, cord blood from the same person, that can be used to treat cancer, and these are important sources of stem cells. So in personalized medicine, at least in certain types, certain specific conditions, I think stem cells play a very important role.
I hope that question is clear to Meghma. Next question is from Sebin Jolie. He's asking that how much does computational softwares help in drug development and the success in diagnosing and reducing side effects in current drugs? Does it have advantages over real-life experiments done using traditional medicines?
Okay, that's a very long question. Let me recall and try to answer what I remember. Your question was about computer programs, computer applications. What was the actual word you used?
Computational softwares help in drug development.
Yes, computer softwares are used in drug development a lot. They can be, for quantitative structure-activity relationships, like I told you. There can be combinatorial chemistry work, meaning storing up, this databases that have known functions of millions of molecules, and you can use computational softwares to sort of filter through that and get the right information. In modeling, a lot of computational softwares are being used. For example, if you want to understand the toxicity of a molecule and you want to sort of simulate that in a computer model without giving it to humans, for example, because you don't know if it is highly toxic and you want to have an idea how toxic this may be. So before you introduce into human body, you could use computational softwares, and computational softwares are used for that purpose. Then there are computational softwares that help you design molecules, based on the quantitative structure-activity relationship principles that I just mentioned. It will tell you how you need to modify a molecule in order to have a certain property. So there are many, many different areas where computational softwares are being used in drug development. It is an extremely important and highly valuable tool. It can be used for any kind of drug discovery, including traditional medicine discoveries. But your question is a very, very general question. And unless you have any specific questions, I won't be able to give you a specific answer. This is a very general answer.
He has also asked that does it have advantages over real life experiments done using traditional methods?
Yeah, advantages over real life experiments in the drug development, yes. But you will not be able to release any drug into the market purely based on an in vitro or in silico computer exercise. At some point, it will have to be tested in humans, and that's why we do clinical trials. But the advantage is that, before you are actually able to do that, you can sort of get an idea about how safe the drug is. And if it turns out to be something very bad, then you probably can tweak the molecule and make it maybe less toxic using this computer software as a guiding principle. You could do that. Yes, that's the advantage. But it's not one over the other. You won't be able to replace in-human testing with computer testing. Did I answer your question?
Yes, sir. Thank you.
Okay, another question from Mini Suresh. She asks that, please tell about advantages and disadvantages of nanoparticles in medicine, how the appropriate metal is selected for making a nanoparticle for a particular plant extract to combat microbial diseases.
How nanoparticles are used to treat diseases, and you have some specific questions towards the end of the question, but let me start generally, because the specific antimicrobial diseases, that's too specific a question. But nanoparticles, let's say for example, nano gold particles are used for cancer therapy. So as a guiding principle, how do you choose which metal? Obviously you choose a metal that is inert. You choose a metal that has the right kind of property that you want to have. And I'll give you one example. In hyperthermia, for example, one of the very fascinating approaches to treat cancer is using gold particles. Gold is a metal. Gold nanoparticle has an advantage that it doesn't react with anything. It's an inert metal, and it absorbs heat. So what you do is you inject gold. Actually, gold, elemental gold is good for you. So you put gold in the system, in the systemic circulation. And what happens is the gold particles will go everywhere, and it will also go to the cancer. But the cancers are avid eaters, because cancers are always hungry. They're dividing very fast and they need a lot of food. So cancers will take anything that come their way. So cancer absorbs all of these gold particles into it. So after some time, you will see that majority of the gold particles you delivered into the body is now concentrated in the cancer. And then what do? You have a metal or metal particles inside the cancer tissue, And you know that if you heat metals, metals get really, really heated up. So one approach was, and it's a brilliant approach, was to sort of locally heat by infrared heating technologies. You heat the tumor, and again, it's only applicable in certain types of cancers. And using hyperthermia machines, you can actually very precisely heat the tumor, and then the gold particle will become highly heated up and will destroy the cancer cells. And because the gold particles are not taken up by anything in the surroundings, they won't be affected. This is one example of using a nanoparticle for a treatment. But different nanoparticles are used for different purposes. For example, silver. Silver has antimicrobial properties, as you know. So you would pick silver for that kind of purposes. So choice of metal really depends on what you want to do. Did I answer your question, or did I miss anything?
Mini, hope that question is clear to you. Okay, next question from Neenu P. Raju. She's asking, sir, there are so many microbes associated with plants. So do you think they have any particular role in medicines made from plants?
He will have to give me an example. I don't know enough about it. It's not, ethnobotany and botanical extracts and plant-based medicines are not really my field.
Okay, Neenu, are you online? You can ask the question directly.
Are you talking about any particular microbe?
Not exactly. She's not asking about any particular microbe.
So the question is, will they have any role in plant-derived medicines for antimicrobial purposes?
Yes, yes.
Yeah, I mean, I would think so. But then again, if she has a specific example, that would be helpful.
Okay. Okay, we'll go to the next question, sir. Noroz Hassan has a question. Apart from gene editing, what are the other significant advantages of CRISPR or Cas9 system?
Apart from gene editing is the main application that we talk about when we talk about CRISPR-Cas. It basically is a gene editing tool, and it is used differently in the bacterial system. We have made it programmable, and that is primarily used for gene editing. And I'm not aware of any other application for that.
Okay, I hope all the questions are clear to you. Anyone you want to ask question?
Hello.
Yes, you can ask.
Good morning, sir. I'm Dr. Radhe. I want to know, sir, in Petri dish, grow cancerous cells, and we put those cancerous cells in healthy human body that can react or metastasize in healthy human body.
You grow cancer cells in a plate and then you put it back into the human body. Will it grow cancer? Maybe not.
Okay. Sir, why, and can you please explain this thing about?
Well, it's a very interesting question, though. Again, our immune system is very powerful in finding any foreign bodies and fight it. That is why tissue grafting is problematic.
Sir, immunocompromised like HIV and TB patients, it can grow about and metastasize other organ.
Yeah, that is what I was getting to. So if your immune system is extremely compromised, then it is possible. Yes.
Okay, thank you, sir.
Anyone from the participants? Any questions?
Yeah. Can I?
Yes.
So I tried anti-cancer silicon through three cancer cells, basically breast cancer and colorectal cancer and also lung cancer. When I did in silico studies, the bonding energy was way higher. In vitro, the IC50 values I got were not up to the mark. In silico studies said when I did the simulation and all, it was very good. The drug I tried to extract and test it. It had a very good high bonding energy and all, delta G and all were very good. But when I did in vitro studies, the IC50 values were not up to the mark. Only the lung cancer was quite fine. The other IC50 values were too way higher.
Yeah, no, this is an interesting question, and I can think of my old days, my previous life, spending many years in the lab setting, doing similar kinds of things. I can only think about a few things. Again, first of all, any modeling is only modeling; it is not perfect, as you know.
True.
So in silico models have their own limitations, and then you take a cancer cell. The cancer cells are not all made the same way, meaning they are not all made equal. In the sense that the same cancer cells that you use in your lab and I use in my lab, for example, HeLa cells, may not be the same. Cancer cells mutate over time. Cancer cells evolutionarily change over time. So the population of cancer cells that you're testing could be something else. They acquire resistance to so many things. It depends on what you tested. For example, let's say you tested something that is also present in something else, during the course of maintenance of the cell line that the cell line was selected for or selected against. So you know where I'm going with this. So if the cancer cells have, for whatever reason, acquired mutations that give them or impart them resistance to what you are trying to do, the pathway or the particular molecule, then you won't find any effect. The third thing is the extract that you're using. Again, extracts are tricky. You cannot define an extract, right? Unless it's purified. So in your simulation, I don't know how you did this. How would you define an extract for simulations? You will have to know a lot about the exact composition of the extract in order to be able to do that. In real life, unlike in computer modeling, the extract will behave the way it is. In the modeling, it may have behaved better because you put in wrong information. Did I miss anything? Did I answer your question?
Yeah.
So the question is, if you purify the active and then you do the simulation and do the experiment, you should be able to see more consistent results.
Okay. Thank you.
Thank you.
Another question from Cindy Boy. How brain tumor can happen although there is no cell division in mature neuron cells?
Yes, in brain, the normal healthy neurons don't actively divide, but there are dividing tissues in the brain, and those are the tissues that actually cause brain tumors. The neuronal growth can also happen under certain circumstances, but the brain is not all one type of cells. There are many different kinds of cells.
Is there any questions? I hope. Any questions from the participants? Okay, there is a question from Sebanjoli again. So what cells were shown in the cell culture you showed in the slide? What cells
What
were shown?
Yeah, this was a movie that I made in 2006. I don't exactly remember what kind. I mean, this was a mammalian cell line. It was a cancerous cell line, but I don't exactly remember which one was used. I have made, movies of all different kinds of cell lines.
Okay. Any questions? Okay. Another question from Nupur. How do we perform ligation with blunt ends?
That's
a common question.
That's a very technical question that I don't even, I wouldn't venture to answer that, because again, that was a long time ago that I did those kind of things. I don't do those kinds of things anymore.
Okay. Okay. Hope everything is clear. Yeah, one more question. When from Cindy, when oxygen is normal, VHL, what regulates? Cindy, are you online? Can you ask the question directly?
When oxygen is normal, what regulates VHL? Is that the question?
I hope so.
Well, the prolyl hydroxylases that I mentioned hydroxylates HIF1 alpha, and that is the tag for VHL to bind and direct the HIF1 alpha for destruction. So the regulation happens at the prolyl hydroxylases level. If the oxygen is not there, then the hydroxylation doesn't happen, and then HIF1 alpha is not tagged and it's not degraded, so the HIF1 alpha is present. That's the way regulation works.
Okay. So
it's like we put our garbage out for pickup, right? If you put the garbage in a designated garbage container, then the garbage pickup guys will come and pick it up, because it's targeted, it's tagged for disposal. But if you put your garbage in a nice looking box and leave it outside, it won't be taken and destroyed, because it's not tagged for destruction. So same way, HIF1 alpha protein is, when there is a lot of oxygen, HIF1 is not needed. So these prolyl hydroxylases will sort of label the protein with hydroxyl groups, and that is actually a signal for it to be taken away and destroyed. I hope I answered your question.
Okay, so it is now long 2 hours now. Still, you have any questions? You can mail me, and I will.
Yeah, maybe there was one question that just popped on the screen. Can we take a look at it?
Yeah. HILA cells are immortal, but we have never heard of any cell which is immortal. What is the difference in it?
Say it again.
HILA cells.
Yeah, HILA. HILA or HELA? I think the right spelling is HELA.
Okay, HILA cells are immortal, but we have never heard of any cell which is immortal. What is the difference in it?
Well, immortalized cell lines are cell lines that can continue to replicate. In normal, so think about a normal cell will not continue to divide and replicate forever. It will divide for 25, 30 times, and then it senesces. It sits there in a state of senescence. Immortalized cells are cells where there are some oncogenic activity, E1A or E1B 19K, P53, these are examples. Those oncogenic pairs, if you put that into the cell, then this cell becomes immortal. So normal cell, an immortal cell. Immortal cell only means that it just goes on forever, it just divides. And then there are transformed cells. Transformation happens by oncogene activations. That's how cancer forms. And that is a very aggressive, but it's just not immortalization. It is now able to grow into big tissues and they are much worse. They can separate, escape, and they can go to other parts of the body, metastasis, and they can grow in other parts of the body. That's a transformed cell line. So these are the differences between 3 different types of cells. And your question was not very clear to me. You said HeLa is immortal, but we haven't heard of immortal cells. The question, I didn't completely understand the question, but I hope I answered your question.
Thank you, sir. So, next question from Cindy. What conditions required to grow cancerous cells?
What conditions can grow cancer cells? You need cancer cells are very good to grow because they are transformed. They have the genetic mutations in them that will help them grow. But the cells, all of them have some requirements. They need nutrients. They need growth factors. Growth factors are factors that are released by the neighboring cells. So they want to feel like there is somebody with them. They're social beings also, just like us. So the presence of other cells around actually keep them happy and they grow better. So when you try to grow them in a plastic plate, you add things like growth factors into them. So nutrients and growth factors, and then right temperature. And that's it.
There's another question from Shravanti. For the treatment of cancer, it is very efficient, is it very efficient or not? Still in a dilemma. How do they help? Please, can you explain, sir?
What was it again? The starting?
Yeah, stem cells are being into practice for the treatment of cancer, but it is very efficient or not?
Oh, I think I answered that question. It's not a one-stop shop. It's not a solution for all cancers. In fact, in some kinds of cancers, after the proper therapy is done, stem cells may be helpful. Because, think about it, your tissue has already changed. And you have cells that are transformed and powerful enough to grow and distribute to different parts of the body and metastasize. So because of that, unless you get rid of those cells, you're not completely out of cancer.
Okay, I hope that.
Yeah, but if you actually somehow kill all of them and take out the tumor, then you can. You also take out some of the normal cells, and then you could rebuild that tissue by using stem cells. At least theoretically, it's not that easy, but at least in principle, it is possible. But it's not that complete of a solution.
Okay, she has asked again, how do you build up a mechanism to prepare drugs in treatment of cancer? Is there any special formula?
I don't think so.
Okay.
That is, yeah. Okay, please.
Yeah. Any further question? Anyone? If any question, you can mail me.
Yeah, I also wanted to, in fact, I had it somewhere here. I forgot to show that. But just thank you. And this is my email, just in case anyone wants to write to me, I'll be happy to answer your questions on anything.
Yeah, okay. So last question from Mini. Why certain drugs which are present in the market are eliminated from the market all of a sudden without any valid reason?
Well, it's never without any valid reason. The medicines are withdrawn for definitely because of some reason that you may or may not know. The companies are, like I said, expected to constantly monitor the safety of the subjects. And they do get a lot of inquiries from the users of the medicine and also the healthcare providers like doctors. They have a lot of data. If there is something wrong somewhere, usually that results in a withdrawal of the medicine, not otherwise. Because why would someone take a medicine that is working without any problem and remove it from the market? That doesn't make any sense. Again, this is a very general question, and if you have any specific examples, then we can talk about it.
I hope that question is clear to many.
Excuse
Okay.
me, sir.
Yes.
Yeah, please.
So I'm Nupur. So I asked you about the ligation of blunt ends. So actually, I am a third semester microbiology student, and this is about sticky ends. We all know, like how could we ligate them, but about blunt ends. I have a very vague knowledge, and because obviously this coronavirus has because of that the colleges are closed and we don't have any practical knowledge. So if you could just give me because on the net I was not able to get a appropriate
Yeah, so
answer.
what I can do is I can look back to what I have done 25 years ago, 25 years ago. And there definitely are blunt end ligation techniques that I can send you. And if you just send me an email, I can give you more details on that. That these are, very easy now. You can get tools to do all of these things. So I can send you those information if you want.
Okay, sir. Thank you, sir. I'll definitely mail you.
Okay, dear participants, you can take down the email ID of Dr. Robin Matthew. If you have any further questions, you can mail him and ask him. And we'll end up the session for now. It is almost 2 hours long session. Thank you, Dr. Robin Matthew. It was a very exciting and a very informative talk. Thank you so much. And thank you so much for sparing your valuable time with us. Thank you once again. Thank you, dear participants. Thank you all. One more thing. I invite one of our student, Minna Nasser, to share a few words, to share the vote of thanks.
A very fine morning to one and all. It's my pleasure to deliver the vote of thanks on behalf of the Department of Microbiology and Biochemistry, SB College, Changanasheri. Firstly, it's my honor to thank our speaker for the day, Dr. Robin Matthew sir, for his very valuable time and effort. Your lecture has given us a deep insight on the topic. Thank you very much, sir. I would like to express our gratitude to our principal, Dr. Jacob Matthew sir, and our director, Father Teddy, for their all-time support. I thank our HOD, Mrs. Gigi Jacob, our coordinator, Mrs. Bibi Sarah John, and all the teachers and students of the department. I thank all the technical assistants who have helped in making this program a successful one. Last but not the least, I thank all our participants for your support and cooperation. Once again, I thank you all.
Thank you, everyone. Thank you for the questions. Really appreciate your asking questions. And I'll be happy to answer them. If you send an email, I'll be very happy to answer your questions.
Okay, dear participants. See you next time.
Thank you.